Okay, so our speaker tonight is Dr. Simon Mitchell. Dr. Mitchell studied computer science and maths at university before completing a PhD in the new field of systems biology. He established his own research group at BSMS in 2019, which focuses on tackling blood cancer using systems biology. So at that point, I'll hand over to you, Dr. Mitchell.
Thanks so much for the intro, Zach, and hi, everybody. Hopefully I will share my screen right now and you can see that. So I'm going to talk to you about what I think the future of healthcare will be. And I'm a cancer research lecturer at Brighton and Sussex Medical School. So I have a research group that do cancer research, and I'm going to talk to you about just a little bit of the cancer research that happens at BSMS. And whether we can use computers to try and improve treatments for cancer. So what I'm going to cover is this term systems biology, which you maybe didn't even see before you looked at the title of this lecture, and answer the question of how we can simulate life with systems biology and whether that helps us treat cancer. And then I'll give you what I think my vision of the future of cancer treatment might look like. So this is to give you an idea of some of the exciting research that happens at BSMS, and that hopefully if some of you join us and become students at BSMS, you can take part in some of this research as well. So the first thing I want to get you thinking about is this term systems biology, which before I came to university, I'd never heard of before, not a term I know. And I discovered it when I started doing my PhD. It's quite a new term, but not that new because there are textbooks on systems biology, and here's just a few of them. And I want you to just think about whether you know what this field might be. So it's a subset of biology. Maybe you've heard of it before, maybe you haven't. Maybe you can try and guess what it might be. Some of these textbooks have some clues on the cover. And just while you're thinking about that, I'll show you that there are, in fact, institutes of systems biology. Here's one in Washington, the ISB, the Institute for Systems Biology, a whole building dedicated to this field of research. And, you know, the field of systems biology has an institute. There's a receptionist at the Institute for Systems Biology. I think we've made it. We've got a building. So just give you a couple more seconds to think about what on earth it might be before I start to tell you. And I think my favorite definition of what systems biology is comes from Harvard Medical School. So if you go to the Institute of Systems Biology at Harvard Medical School, on the front page of their website, they have this. And I love this. I think this really summarizes it for me. So in systems biology, we use the tools of mathematics and computation because biology is too complicated for the unaided human brain. So that's why I use systems biology, because biology is way too complicated for anyone to understand on their own. It's too complicated for me, and I've been thinking about some of this stuff for a long time. Biology in general, and then medicine and understanding drugs and how to treat people, it's fiendishly complicated. And some parts of biology are just too complicated for any one brain to comprehend without the use of mathematics and computation. And then you might start thinking about what complicated means. What do I mean by biology being too complicated? Well, it's complicated because of all the interactions, all the things that interact with each other. Within a cell, within a population, within people, when medicines interact, on all of these scales, there's just so much complexity created that it can be very, very hard to know what's going to happen predictably. And when you're trying to treat somebody and know that they will respond to a drug, it helps to try and understand those interactions and know how a patient might interact with a drug, how they might respond to a virus, those sorts of questions. And a time when you might have seen systems biology recently is on the news. This is me scrambling from my sofa to grab my phone and take a picture of the news because I saw systems biology on the news. And when the COVID outbreak happened, there was a lot of complexity that we didn't understand. We needed to quickly understand how susceptible people might get infected, how many people might be exposed to COVID, how many people might currently be infected, and how those things might progress over time. And that's what these equations are trying to do. The S here is susceptible. The E here are people exposed, and the I here are people that might currently have an infection. And it gets a bit more complicated than that, but we were using these equations to try and predict how the pandemic might progress. And that's exactly what I do, but not necessarily with pandemics, but with cancer. And so I think the best way of really explaining what I do and getting those ugly equations off the screen is to think about my favorite protein. And that's right, when you reach my stage of your career, you might have a favorite protein too. My favorite protein is NF-kappa B. It's beautiful. I'm sure you'll all agree. Here it is. Here's its structure. There's two parts to it. It's binding to DNA. A lot of work has gone into figuring out this complex structure of this protein. But for the purposes of this talk, I'm going to reduce it down to an egg. Well, to two eggs sat next to each other, because it's two proteins. And I'm very sorry to all the amazing scientists who did all that structural work. But for now, all we need to know is that it's two proteins bound together. So I've reduced my favorite protein down to these two eggs. And just to give you a bit of background about where this protein came from, it was discovered in the lab of David Baltimore. He was a Nobel Prize winning scientist even before he discovered this protein. And him and a postdoc in his lab, Ranjan Sen, discovered that there was a mysterious protein in the nucleus of cells, so right in the middle of these cells, that was able to bind to certain elements of DNA called the kappa enhancer. And they found it in a whole bunch of different cells, but mainly in B cells. So they called it nuclear factor kappa B. It's a nuclear factor in the center of cells that binds to these kappa elements in B cells. So that's where this name came from. And so that's when my favorite protein was discovered, my favorite pair of proteins, really. And then a couple of years later, they discovered that there's another protein involved that actually inhibits NF-kappa B. So it stops NF-kappa B from binding DNA, inventively called an inhibitor of NF-kappa B. So this I-kappa B comes along. So now my favorite protein, my favorite eggs have an inhibitor that's involved as well. And that was from way back in 1988. The previous paper was 1986. And then since then, people have kept working on these proteins and found they're really, really important. In fact, if you type in NF-kappa B into a search of the scientific literature now, you'll find 800,000 results for this little egg-like protein discovered in 1986. And what are all those papers about? Well, it turns out that it's a very, very complicated protein, because it's not just these two proteins. It can be any one of a number of proteins, and those combine together to make a whole bunch of different combinations. These two proteins can actually be any one of these proteins. And then this inhibitor up here, we've discovered so much that we're into the Greek letters now. There's alpha, beta, epsilon, delta. You need to know your Greek alphabet to understand all the different inhibitors that could be bound to NF-kappa B. So there's loads of complexity that's been discovered about this protein. And when it's not just about the actual protein itself, but it's about how it's regulated. If we try and summarize all of that information in a picture, you end up with a crazy spaghetti diagram that looks like this, that's so hard to understand. It's so complicated. And when presented with something like this, we have two choices: that we either think this is too complicated, we're never going to understand it, or we try. And if you look at the top result in the scientific literature for NF-kappa B, in the papers that other people have done, you'll find they all talk about cancer. This protein is really important for cancer. This protein, when it's activated, makes cells survive too long. It makes cells divide a lot. Cancer divides a lot. Cancer survives too long. This protein really is at the center of being able to treat and understand cancer. So the option of just ignoring all this complexity goes away if we want to try and treat cancer better. So then I go back to what Harvard Medical School said, that when biology is too complicated for the unaided human brain, we need to use the tools of mathematics and computation. And I would argue this diagram is too complicated for the unaided human brain. I've thought about this protein for a long time now, and I still couldn't tell you the way all these things might interact in a real cell. And that's where using maths and computing can come in. We might ask questions like, what happens if this alpha protein, this little alpha protein here, becomes mutated? Or what if the beta protein becomes mutated? Because I've spent a long time thinking about this system, I might know that that might make this go down, which might make this go up, but I don't know what that's going to do over here. I don't know what that's going to do down here. It very quickly becomes impossible to predict the way all of these proteins will respond to these mutations. And these mutations are really important because they exist in cancer patients. There are patients that have these mutations. So we need to be able to understand how this network is going to respond. And my approach to understanding this is taking all these interactions and encoding them into a computer. That might sound a little out there, but I'll show you how we do it. It's not that complicated. We encode these interactions into a computer, and then we ask the computer what's going to happen if those mutations exist. We can simulate the response of a cell to those mutations, and we don't have to guess what might happen or do a load of experiments. We can instead just ask the computer, if that mutation exists, what might happen? And I'll show you how we do that in a second. But that's exactly what was done by Alex Hoffman. He was another postdoc in David Baltimore's lab. And what he did was take these interactions, encode them into a computer, and then ask what mutations might do to those proteins. And here we have some mutations. Here we have some different mutations. And we're looking at what NF kappa B is doing over time. And especially this one is interesting. The computer's predicting that it's going to go up and down, it's going to oscillate. This looks like something you might see out of a sound wave, out of a wave pool. This doesn't look like biology. This looks very strange. But this was the prediction that the simulations generated in response to these mutations. And when you get weird predictions like that, you can do some experiments to test them. So that's exactly what they did. They mutated those genes in cells. And look, this is the band for the protein. It's going up and down and back up again, and you can quantify that. And on the right here, we have the experiment. It's going up and down. It's just like the prediction. This up and down oscillation here looks exactly like this up and down prediction over here. So this crazy behavior that was predicted in the simulation really does exist in the lab. And only through simulating this are we able to understand what's going on. And that's when I started thinking about this. Alex Hoffman then started his own lab. I became a postdoc of his over in the University of California. And I took the same approach to try and understand the way our cells will respond to bacteria, responding to viruses, responding to other chemicals that are secreted by our cells, generating predictions, and then validating them with experiments. And this is just one example we published where indeed the simulation predicted that the cell would respond differently to viruses than it would to bacteria, and that a certain chemical that was in our blood would enhance one of those responses and not the other one. And then we did the experiment and saw the same result. So the simulation is really helping us understand the way our cells are responding to the various things that cells respond to. So this field is not small anymore. This is a huge field. So this is a picture of a bunch of systems biologists at a systems biology conference. This one was in 2019 in Japan. I was recently at their latest one that was in Berlin. And it's a growing field that's really exciting. And I think I've shown you a few sort of old white men involved in the history of this field, but the future is looking very different. And these are just some of the people that really excite me in systems biology. Mariko Okada, Jennifer Euler-Yaniv, Stacey Finley, and Jana Wolf. They're all doing amazing work on cancer, on immunology, on understanding how our cells respond to viruses, all with a computational and mathematical approach combined with experiments. So when I say we're going to simulate biology, and I said we're going to take all this information and put it into the computer, what does that really look like? What does that mean? Well, we were asked that question quite a lot, so we wrote a textbook chapter on it, and this is really just a step-by-step guide of how to simulate biology. And I tell everybody that it always starts with a picture. So that's the universal language that we all speak. I did a computer science and maths degree, but I can speak to someone who's done a biology degree or a medic who's seeing patients every day, and we all speak the same language in pictures. So I often sit down with experts and draw pictures to try and understand the way things interact. And the pictures will sometimes look like this. So I'm taking my favorite egg-like protein, NF-kappa B, here, and that inhibitor that was discovered shortly after. And this might be the network that I'm trying to simulate. These two things can bind together. They can interact together to make a complex, or they can unbind again, which is why we have arrows going one way, arrows going the other. And it really just starts with a picture. And if you get the picture well enough, if you do the picture accurately enough, that's half the work done, really. The next step, and this will be the only equation I show, is to turn that picture into an equation. And this equation might look scary, but really all it's saying is that if we want to understand how much of this yellow protein there is in the system, we need to know that it's going to go down when it moves into the complex, and it's going to come back up again when the complex breaks apart. So we have a negative sign here as they go down, and we have a positive sign here as it comes back out. And that's all this equation represents. When we do that for each interaction in our network, and we then turn the equation into computer code, some of you might have heard of Python code, some of you might even have written a little bit of code. Don't worry if you haven't, but we can convert this into code. We use a language called Julia, but it's a lot like Python. And you'll see that this computer code looks a lot like this equation. We have a number called K on, we have a number for I kappa B, a number for NF kappa B. We just line by line turn the equation into code. And then that's it. We just pass this equation to the computer and ask it what will happen to our system. We don't have to sit down with a pen and a paper and solve the equation. I wouldn't be very good at that nowadays, to be honest. All I do is pass it to the computer and ask it what will happen. And we get these predictions, virtual experiments where we can see how things might change over time. And when we've done a virtual experiment, it's always good to compare it to a real experiment to see if we got it right. So I showed you this example earlier, which is an example where we did get it right, because the simulation on the left from the computer looks a lot like the experiment on the right that was done in the lab on real cells. But it doesn't always go right. So we ask the question, did we get it right? Does our simulation look like our real biology? And the answer can be yes or no. And I'd love to say that we get it right every time, but that we don't. We get it wrong as often as we get it right. If we get it wrong, though, it means something was missing from that picture. When you show the picture and you show the things that interact and you sit down with an expert and draw the pictures to try and understand what's happening, if you get that picture right, the simulation will work. And if the simulation doesn't work and doesn't match the experiment, it means something was missing. And I've published papers where that has happened, where I've run a simulation. It looks nothing like the biology. The cells in the lab are not behaving like the cells in the computer. And then we go back to the picture and we think, what might be missing? And we try a few more simulations. We do a bit more lab work to try and figure out what was missing. And in this paper, we discovered a really important interaction for how B cells decide what to do in response to an infection. And we wouldn't have found that if the simulation had worked first time. The simulation didn't work because we didn't know something. So when we get it wrong, it's good news, at least if we want to learn more about how cells behave. And then if we get it right, we've got a cell that works, that is a virtual experiment, that in a few seconds, I can say, I can ask it what will happen over time, or what will happen if I add a drug to the system. And in a few seconds, without going to the lab, without growing up cells, without spending money on reagents, I can get a virtual result that will tell me what will happen. So when it works, it's exciting. When it doesn't work, it's exciting. The real question is, how on earth can we use this approach to understand cancer? And that's what I set up my lab at BSMS to understand. I started a lab and I've got a growing team all focused on using this approach to understand cancer here at BSMS. And specifically, I'm looking at cancers of B cells. You might have heard of B cells. If you haven't heard of B cells, they've been very important in your life lately because they produce antibodies. They fight infections. And so we've all been hoping recently with pandemics that we've got enough antibodies to fight these infections, and enough B cells producing enough antibodies to fight these infections. So the thing about B cells is they have to divide very rapidly. You need a lot of B cells very quickly in response to an infection. If you get COVID, you need a lot of B cells producing a lot of antibodies to fight that infection as quickly as possible. And it's the same with lots of different infections. But they always exist in this careful balance, because having B cells dividing out of control is also not good for you. You don't need too many B cells. You don't need not enough B cells. You need just the right amount to fight the infection, for the infection to go away, and then the B cells need to die and stop producing antibodies. If you produce too many antibodies to too many of the wrong things, if your B cells are dividing too much, you can get autoimmunity, where your immune system will fight itself. Or the real far end of the spectrum, which I study, if your B cells don't die when they should do and divide too often, this uncontrolled proliferation, that's B cell cancer. That is the B cell lymphoma that I study and that I want to use computers to understand. So the most common type of B cell lymphoma is diffuse large B cell lymphoma. We'll shorten it to DLBCL. It's a cancer of these B cells where they're dividing too much and they're not dying enough. It's the most common lymphoma in the UK. There's about 6,000 cases a year, and those case numbers are increasing because we have an increasingly elderly population. People are surviving long enough that more and more cancers are being picked up. And currently, people are treated, people will arrive at the doctor's, maybe they'll have a lump on one of their lymph nodes, and the doctor will take a sample from that lymph node. The patient might have night sweats, for example, they might not, but the doctor will take a sample from that lymph node, which will go to a diagnostic team who will look at the cells and try and figure out whether it is a B cell lymphoma. If it is, then they'll receive a cocktail of drugs that we call CHOP. It's a chemotherapy regime that is the standard of care for everybody. And it's quite a good treatment. This graph will show you how good it is. And this graph might not look like much, but this was a real breakthrough in the field of B cell lymphoma work, because this graph shows what's your chances of being alive over time, over the years after being diagnosed with diffuse large B cell lymphoma. And CHOP, this CHOP is the standard of care, or was the standard of care for about 40 years. CHOP stands for cyclophosphamide, doxorubicin, vincristine, and prednisone, but you don't need to worry about that. It's a cocktail of drugs designed to hit cells that divide too much, which is exactly what we need to counter lymphoma. And as you can see, for the 40 years we were using this treatment, you had about a 50% chance of being alive after 4 years. And then R-CHOP came along, which is added a drug called rituximab to the mix. Rituximab is amazing. It targets B cells specifically. So rather than broadly targeting cells that divide a lot, it started targeting B cells with an antibody that binds only to the surface of B cells. And that really improved survival. This paper came out in 2010. R-CHOP started being discovered as a better way of treating people around the year 2000. And this curve has improved a little bit now, to the point where about 60% of people are cured with R-CHOP. So the treatments have got quite a lot better, but 60% isn't enough, really. If you're in that 40% that don't get cured by the current treatment, you'll have a relapse disease. And for those people, the prognosis isn't good. I've been to lots of conferences where graphs like this are shown for people with, for when the disease relapses. And really, the x-axis here is in months. So you've got a 50% chance of being alive at 6 to 8 months. It's really not great, the prognosis for a relapse disease. And there's been a whole load of work. There's new trials all the time trying to move this curve, some with some success, thousands of people enrolled on trials. but we're still struggling. And for those 40% of people that for whom R-CHOP doesn't work, we really do need something better. And that's what my lab is working on. So just to give you a timeline of the treatment for DLBCL, R-CHOP came along in the early 2000s, as I said, and for 60% of people, that was good news. But since then, we've had no changes in the standard of care. But in that time, we've learned a lot about the disease. It's not like people have stopped studying it. There's been thousands of papers. We first discovered that actually, it's not just one disease. It's a couple of subgroups of disease, so-called ABC, diffuse large B-cell lymphoma, and GC. They have different prognoses. One is more aggressive than the other. They have different biology. We've learned how important my favorite protein is in DLBCL, that they often have high NF-kappa B. We've learned through the advent of sequencing technologies where we're able to actually look at the mutations in individual patients. We've learned a lot about what mutations cause the disease. Some are very common in lots of patients and some are quite rare. We've learned about how some of these mutations combine such that having a combination of certain mutations seems to give you a more aggressive disease. And even more recently, we've learned that actually there's two subgroups of patients in the disease, might be more like 5, 6, 7. It's really an umbrella of different diseases with different biology. And you could look at any particular patients that have been sequenced and look at their mutations, and you'll probably find it's different from any other patient. So is it really a surprise that giving them all one therapy doesn't work for a large proportion of them? I don't think so, and I think that's why we need to move to a better way of treating these patients. Another way to illustrate all of this stuff we've learned about DLBCL is with my favorite egg-like protein, NF-kappa B here again. We know it's high in B-cell lymphoma. It's often involved in B-cell lymphoma. This is a diagram of just some of the ways that NF-kappa B is regulated. It's fiendishly complicated. This is the surface of the cell. There's receptors on the cell, and then they might sense danger signals in your blood, and then that can give rise to proteins activating more proteins, which activate other proteins, and ends up with NF-kappa B moving to the middle of the cell. So we've learned a lot about the biology of this system while we've been using the same treatment for everybody. And we've also learned a lot about the mutations that can exist in this system, all the way from the top, from the outside of the cell, all the way down to the bottom inside the nucleus. You can get mutations that exist in patients that cause cancer, that cause B-cell lymphoma. And we know a lot about these mutations and how they differ between different patients. We've also, that's the bad news really, but the good news is we've got a whole suite of tools to try and tackle this problem. We've got inhibitors that could potentially be useful drugs that target this pathway from the top to the bottom. So there's mutations from the top to the bottom. We have ways to target the pathway from the top to the bottom. And the real challenge in DLBCL is getting the right drug into the right patient. Can we know that this drug will work in this patient, this drug will work in another patient? And currently there's no real way of knowing. In our lab, we knew that NF-kappa B had been studied quite a lot in this disease, but everyone was just generally thinking about whether it was high or low. And we thought with all this variability that we found in patients, whether that's enough to understand the disease. And so we measured it. We measured just the different components that could go into NF-kappa B. So my favorite egg-like protein could be made up of this REL-A protein or this REL-B protein. And we wanted to know, do patients look the same? Do they look different? Can we try and find a way of getting the right drug into the right patient? And how different are these patients? Because nobody really knows. We expected to see something like this, where our healthy cells would all be low in NF-kappa B. So they're low here and they're low here. They're down the bottom left because they're low in NF-kappa B. And we knew from all the other studies that cancer cells and lymphoma cells would be high in NF-kappa B. So we thought they might be up here where they're high in both of the proteins we were measuring. And so a postdoc in my lab, Eleanor, along with Andrea Pepper, went into the lab and measured these proteins in different lymphoma cells. And this is recent work that we've generated, so it's really exciting to share it. And we found that actually every patient is different from every other patient. So down here we have our healthy cells, so that's good. Indeed, the healthy cells are low, but the blue lymphoma cells versus the green versus the light blue versus the red, every different lymphoma cell we put into here looks different. We take different groups of lymphoma cells, figure out how much of these things they have, and each one looks unique. So we're really starting to think that each patient has a more unique fingerprint of its disease. And is it really any surprise that these blue lymphoma cells might respond differently to a drug than these green lymphoma cells? They're very different cells. This isn't really just one disease. It's a very broad, heterogeneous, diverse umbrella of diseases. And so another way of looking at that is by taking patient cells, taking a biopsy from a patient, putting the cells in a dish and treating them with some drugs. And here the patients are treated with 3 different drugs that all target cell death. They try and make the lymphoma cells die. And on the left is one patient's cells, on the right is another patient's cells. This is from some collaborators of ours. And in this patient on the left, the blue drug works very well. Lower on this graph is better because you've got fewer lymphoma cells left alive. We're trying to kill the lymphoma cells. And at quite a low dose of the blue drug, we've killed most of the lymphoma cells. So if we were going to give a drug to this patient on the left, we would choose the blue drug. If we were going to give a drug to this patient on the right, the blue drug would be the worst. The gray drug would be better. And these drugs all try and kill the cells. They all target cell death. But the blue one is the right one for the patient on the left, and the gray one is the right one for the patient on the right. The problem is we don't know in advance which one will work for which patient and how to get the right drug into the right patient. So I think the challenge here is getting the right drugs into the right patients. And hopefully, what my lab believes is that the solution is to use this systems biology approach to simulate the disease on the computer and predict which drug will work best, so that we can in advance know whether the blue drug would be better or the gray drug would be better. So if you remember my NF kappa B picture that I showed you, that we turned into equations and then we turned into code, we did that trying to understand these drugs that target cell death. And the bad news is that those interactions are a lot more complicated, and you end up with a network that looks like this. So Ilias, who was a postdoc in my lab, made a huge simulation that included all of these lines as bits of equations. And it's about a year's work to create one of these simulations. But the goal was that if we can simulate this, because I think this is too complicated for the unaided human brain. I think Harvard Medical School would agree this is a good use of systems biology. But we thought maybe if we could simulate this, we could understand how to get the right drugs into the right patients. So we did that. We turned them into equations. We turned the lines on the picture into code. We put them in the computer, and we brought in a lot of experimental data from collaborators of ours to make sure that the simulation was accurate. And the result is a simulation that represents a virtual lymphoma cell, or a population of lymphoma cells, that we can then simulate and ask questions like, will they respond to this drug? Will they respond to that drug? And that's exactly what we did. We took our virtual lymphoma. We took one particular subset of lymphoma, so one of those fingerprints on that graph, and we said, okay, which drug would be the best for these lymphoma cells? And remember, lower is better here because we're killing more of the cells. The prediction from this simulation is that the red drug would leave very few lymphoma cells alive at quite a low dose. So the red drug would be very effective in these lymphoma cells. The blue drug would be okay. At very high doses, we'd get most of the cells dying, but you'd rather the red drug. The black drug would be useless. And this is the simulation prediction. And we compared that to an experiment and found that actually the red drug needs a very low dose. This small number here means just a very small amount of the drug can kill 50% of those cells. That's what this EC50 means. It means that a small amount of drug can kill a whole bunch of these cells. The blue drug is okay. It can kill some of the cells, but you need a lot of it. And the red drug is useless. So the experiment that was done by our collaborators perfectly matches what we see in the simulation, that we should give the red drug to these cells. So that's good news, but we've only done one of them. And as we can see, lots of different patients respond differently. So we extended this approach to a whole library of cells. We simulated different types of lymphoma cells and found that the simulation says these cells should get the black drug. These cells won't respond to any drug. These cells should get the red drug. These cells should get the red drug. Maybe the blue drug would be okay too. And these two guys should get the blue drug. So lots of simulation predictions that say that different lymphoma cells need different drugs. And then we compared that to the experiments and saw that we were really quite accurate, that the black drug is the right drug for this patient, that a low dose of this black drug would work quite well. In these cells, that they're all quite non-responsive. These cells don't respond to any of these drugs very well. These cells respond really well to the red drug, as do these, but the blue drug's quite good as well, a bit like we see in our simulations. In the experiments here, again, only the blue drug works, just like in our simulations. So if we were able to simulate these cells, we would know which drug to give to which of these cells, which would be so hard to do without simulating, because the network is just so complicated. And even more exciting, we're able to find combinations of drugs that work together. So in these particular lymphoma cells, the blue drug is useless. It doesn't kill any of the cells, really. The black drug is okay. At high doses, we can kill half the cells. But if we combine the blue drug and the black drug to make the green combination, even though the blue drug did nothing on its own, the two together can kill all of the cells. So this is what's called synergy, where a drug works better in combination than you might expect it to on its own. And this is an amazing prediction if this works in the lab. And then we went to the lab, we did the experiment with a few collaborators of mine from the University of Leicester, and we saw the same synergy where the blue drug did not do anything on its own, but when combined with the black drug, the combination was really effective. So we can find combinations of drugs that work well together in the computer and without doing any experiments, and then validate that those computer predictions were right using experiments. So the next steps of this work are really to understand treatment resistance a bit better. The cells that are left after a drug, are they sensitive to a new drug? We're running simulations at the moment to try and understand that. We're getting patient samples, those diagnostic samples that I mentioned, where patients have some of their tumor taken and used for diagnostics. We can take some of that and use it to inform our models. And the long-term goal is really to bring this forward to clinical trials. So if an algorithm or if a simulation helps find the right treatment for the right person, do we improve outcomes for patients? That's my long-term goal and the long-term goal of the research that I do at BSMS. So just to give you, as I'm wrapping up, my vision for healthcare, this is currently how it is, particularly in lymphoma, that, as I said, you get a sample taken, a diagnosis, and then you receive the standard of care. I think if we can use that sample to generate a few more numbers, a few more parameters that tell us more about that patient, feed all of that into creating a model, a simulation of that patient, we can have a virtual patient that looks like the real patient that's in the clinic. In that virtual patient, we can then try a bunch of different drugs that might be available, whether it's the blue drug, the green drug, the red combination, or no treatment at all. Because for some people, if the treatment's not going to work, the treatments are really quite brutal, it might be better not to treat them. And we need to not forget that either. So we can try a virtual drug trial of giving each of these virtual drugs to each of our virtual patients, get a prediction for which drug might work. It might be that the chances of surviving are better with the red combination of drugs in this patient, and then give that information to the doctor who does the prescribing. So then they'll have an option between the standard of care and a personalized recommendation based on the model. I'm not saying that the computers should dispense the drugs. I don't think I'm going to put doctors out of business, but I hope to give them another tool to make informed decisions that are more personalized to the patients. And hopefully that 40% for whom the current therapies won't work will have a better outcome. And that's what I'm calling, personalized healthcare. Really, the outcome is the drug is personalized based on a simulation of that patient. This seems like a lot, and some of you in the audience are future doctors thinking of going to medical school, and I don't want you to start breaking out your algebra textbooks to learn all of this stuff, because I really think when it's implemented in the clinic, it will just be, as the diagnostic sample goes away, you will receive back on your laptop, on a computer in the clinic, some model-informed treatment recommendations. So I don't think everybody needs to learn how to do this stuff that I do. In fact, I hope not, because it would put me out of a job. But I do think learning to code anyway is really important, and we should all be, it's a great skill to have. So I wouldn't put you off doing that. So my vision for the future, if we're here with RCHOP, having been there for a long time, despite all we've learned about the disease, I think the next step of this timeline is a more personalized medicine approach that I've laid out. And with that, I'll finish talking, just thanking the funding for my research, which is from the UKRI and Leukemia UK, a fantastic charity, a whole bunch of collaborators, more than I have time to list, and BSMS for all my support. And I'm happy to answer any questions anyone has.
Excellent. Thank you very much, Simon. That was really, really fascinating. It's really interesting to get an insight into a different area of medicine. So yeah, like Simon said, if you have any questions whatsoever, please do pop them into the Q&A function. We've got a good one here already. What is your undergraduate degree? How did you start?
So I did an undergraduate degree in computer science and maths. It was a joint honours degree, and then I knew I wanted to do a PhD, but I didn't really know what in. And I was looking at the PhDs that were out there in those fields, and they didn't quite scratch the itch I had for impact in the real world. And I was looking around at things I could study for a PhD, and discovered systems biology and this whole field of using computer science and maths I could use the skills I had to hopefully make a difference to medicine. And that's, I didn't do biology in my degree. In fact, biology in school wasn't my favorite subject, but I've had to relearn a lot of biology since then, because I've discovered how fascinating it is later.
Excellent. All the questions are coming in now, Simon. This is good. So here's we'll go with the second one. Does systems biology take account of long-term effects of the drug rather than just the immediate effect, for example, percentage of lymphoma cells?
What a great question. And yes, it can do. And there are whole groups studying tumor evolution. And it's a totally different timescale to what I've been talking about. My work is moving on to look at that a bit more and more. I'm running these simulations where I give a low dose of the drug to the cells and then see what cells are left alive, and then give a slightly higher dose and then see what cells are left alive. So I'm trying to simulate the sort of treatment resistance that emerges over time. But there are whole groups modeling and trying to use systems biology to understand the way diseases evolve. You can look at what cells are present at time point 0 when the patient arrives at the clinic, and you can look at what cells are alive after they've had 12 rounds of treatment, and you'll find they're very, very different. And so the long-term effects can also be simulated, and it's quite a lot of work to bridge those scales from the real molecular all the way to the long-term months, but it's a goal of lots of people in the field. Yeah.
Brilliant. So we'll carry on down the list as I've got them at the moment. Can the systems be used to find which drugs should not be used in combinations as well due to poorer results?
Yeah. So I showed you 2 drugs that work well together, and we did a lot of simulations that mainly showed that the drugs wouldn't combine in an exciting way. You're lucky if you find that 2 drugs combine in what we call an additive way. So if one drug kills 20% of the cells and another drug kills 20%, if you add them together, if they kill 40%, that's great. You've got a combination that's exciting. If they kill more than 40%, you've got what we're calling synergy. if you've got less than that, you've really not got a benefit of combining the drugs. And sometimes the two drugs counter each other out. So we can simulate some of that. What's more difficult and more long-term is if the drugs work on very different things. So often we have drugs that alter the immune system combined with drugs that target cell death. And so then you need very complicated simulations that bring in a lot of detail, and then the predictions can get a bit fuzzier, but we're working on it.
Oh, fab. So our next one, it's just jumped down for me for some reason. What motivated you to go into your career, and what A levels did you take?
Oh, great question. I did maths, physics, and chemistry A levels, I believe. It's so long ago, I'm struggling to remember. And it was the maths, the physics, and computing that really interested me in school. I thought I would have a career in computing. I thought I'd earn a lot of money doing computing stuff, and then realized there's more to life than that, and that actually I'm just so, I find it so exciting to get to study these things, that I veered away. I had some real jobs on the way, which reminded me that real jobs are hard, and the reward that you get from research that could change people's lives is like nothing else. So I knew I wanted to do a PhD, and I did my PhD, tried some real jobs, and thought, no, I'm meant for research, because it's really so exciting to get to come to work and think about this stuff. And the people you get to work with are so exciting. Everyone in my team's amazing. All the people I collaborate with are amazing. If you want to talk to excited, interesting, intelligent, exciting people every day, then do some research. It's amazing.
Excellent. We've got a question here. So what computer program or programs were used for this?
Yeah, cool. So we use a language called Julia, which is very specific for this, but we're one of the only people using it, to be honest. That you can do it in almost anything. If you're interested in learning a bit of computing, I always recommend Python. There's lots of amazing Python tutorials out there, and there are people who do all of the stuff that I do in Python. Python you can learn easily, and it can do incredible scientific computing. There's a lot of different software being used. MATLAB is the other one, but it's expensive. If you really want to start on this, I would pick up a Python tutorial, make some graphs, and download some data and figure it out. And if you find that fun, give me an email. My email's on the screen, and I can chat to you.
Excellent. We've had a number of questions actually about implementation and utilizing this in the real world, so as to speak. So our first one here, how long would you predict the personalized healthcare approach to take from diagnosis of the patient to providing the personalized treatment?
Yeah, great question. And it's important that it's quick, because delaying treatment for cancer is obviously a bad thing. And even if we get better treatments, if it takes longer to give them to the patient, the benefit might go away. So we need to get those predictions quickly. I don't know yet, but we've got clinical trials in lymphoma in the UK that are able to take samples from patients and perform genetic sequencing on those patients. Use that information, give that information to the doctors, and not delay the treatment. So the computing will be quicker than those experiments. So I don't think it will delay treatment. We could streamline it to be hours as opposed to weeks. But I think we can probably get the answers to the clinic quicker than we can get the drugs into the clinic. So I think that won't be a bottleneck.
That's really interesting. So we've got sort of like a follow-up, I suppose, to this. A couple of people have asked, will this be able to be common practice used in the future? When do you think that personalized medicine will be widely available and utilized by the NHS? Yeah,
it's going to be different in every field, and personalized medicine does exist in the NHS. In some cancers, you will figure out whether there is a certain receptor or a certain mutation, and you'll change the drug that you give people based on that. And there's actually some great personalized medicine research happening at BSMS, some of which has been implemented in healthcare systems. In asthma, there's some fantastic research going on where we find that certain drug combinations work best if patients have certain mutations, but they're not using the sort of simulation approach that I'm using, which I think in lymphoma and in cancer research is going to be key. So I think the timeline for what I do to move towards the clinic is probably 5-10 years. It takes 5 years to get to a trial, and then 10 years if the trial goes well. These things don't move quickly, but I do think it's going to be the future. I used to say 20 years, and then some of my colleagues said, no, it's moving faster than that, and I need to be more ambitious. So let's say 10 years before there's a box in the clinic.
And I think this is a great follow-up question for this one, because it's always on everybody's minds. What or where would you imagine the funding for this type of personalized treatment would come from? It sounds like it'd be very expensive. Would it be too expensive to be covered by the NHS?
Yeah, good question. I need to talk to some decision makers in the NHS and convince them of that one day. I think nothing is more expensive than multiple rounds of treatment. And if you look at the cost of treatment on the NHS, the first line of treatment, giving the same drug to everyone, is quite cheap. But then the follow-on treatments can be really expensive. And you might have heard about CAR T therapy in the news. Some of these people will get stem cell transplants. These are hundreds of thousands of pounds, millions of pounds, some of them, to treat a single patient if the first treatment doesn't work. So the best way the NHS can save money really is getting the right drug in the right patient the first time. And most of what I do is about using drugs that already exist, combining them, finding the right person to put them in. So none of this is expensive. The computing is cheaper than an experiment. So I think it'll save money, but I would say that.
These are all really, really good questions across the board. So well done, everyone. Will we be able to use the simulations to account for other variables to do with individual differences, such as comorbid conditions a patient might have?
Yeah, these really are good questions. These are better questions than I get at conferences sometimes. I'm very impressed. Yeah, comorbidities are tough. We have to think about, in the end, I'm boiling down what's happening in a cell to a number or to a bunch of numbers. And I don't know what number to change for age or for frailty. So long-term, yes. Short-term, no. It's very high. There are certain things, comorbidities, that we know what they do. We know about sort of inflammation and the chemicals that might release and how that might change the system that I'm simulating. But I don't know how age and frailty fit into the equation. So there's still going to be a lot of that sort of decision making in the clinic with the doctor, thinking about how well a drug might be tolerated. But our current treatments are very broad. Chemotherapy that targets all dividing cells, your nails and your hair, and that currently we're just trying to stop cells dividing. And we can find more targeted therapies. These drugs that I was looking at that target cell death should be less toxic, more easy to tolerate, and hopefully work better with various comorbidities, because they target very specific pathways in lymphoma cells and hopefully not in the healthy cells for the rest of the patient. So yeah, that's a real challenge for our field. Well identified.
Another good one here. Do you see your research becoming more cross-disciplinary in the future to co-create solutions? If so, then who are you hoping to collaborate with?
Yeah. So on the screen still is a screenshot from our website, and I describe our approach as interdisciplinary. Mathematics, computing, experiments, medicine. I really want to work with all of these people across these disciplines, and it could go beyond that. There's ways we could engage engineers. I've spoken to people in Canada who are working on nanoparticles to treat cancer, and using more systems engineering approaches. There are people who do synthetic biology, who engineer cells to do certain things. My passion is really to try and impact the clinic with this stuff. So that the main people I work with and collaborate with are doctors. That's why I'm in BSMS. That's why doing medical research in BSMS is so exciting, because the doctors that see the patients, that take those samples from patients, come and talk to me about the disease, about the challenges their patients are having. And then we have a lab right outside that door where we can do the experiments. The computer's here. We have a lot of these collaborations here. And then I do collaborate internationally with other modelers, with other experimental scientists, experts on specific parts of these systems, and clinicians who might have access to patients and samples.
Brilliant. I think we're running a little close to time, so I might have to make this the final one, just so that we have a little bit of time just to wrap up. And it's a good one. Do you have a lot of communications with the hospitals that provide treatment to patients?
Yes, a bit like what I said to the last question. We do work closely with the clinic. We have a hematology research group that I think is very exciting. That's a collaboration between the University of Sussex Life Sciences and researchers at BSMS. And that really spans the gamut from what I do to lab work, and has a number of clinicians in there, the key hematologists in the Brighton hospitals, in Sussex, in Eastbourne. And I'm talking to them all the time, putting in grants, writing papers, trying to understand this stuff with people who see patients. Yeah.
Excellent. Well, thank you very much, Simon. It's been a really fascinating talk. I've seen a couple of messages in the Q&A expressing their thanks from our attendees as well. So I'm glad that everybody's really enjoyed the talk this evening.